Hello everyone. This video is about carbohydrates and this is a continuation of our chapter biomolecules. In biomolecules, we have already discussed nucleic acids and proteins, and in this one, we'll be discussing the carbohydrates. So first of all, what are carbohydrates? Carbohydrates. The carbohydrates, as the name suggests, are the hydrates, meaning H2O, of carbon. So a basic chemical formula for carbohydrates is CH2O. Now it has different modifications that we'll be discussing later. But first of all, what is a carbohydrate and what is its need in human body? So carbs, as we know, are the main source of energy in human body. The energy that we get for doing our daily activities is derived from glucose, which is the very basic unit of a carbohydrate. The simplest carbohydrate that we know is the glucose. Now, the carbohydrates, apart from providing energy, also help in building up the biomolecules of human body. Carbohydrates can be found in the form of either sugars or fibers or starches. So these are the different types of carbs that we find in the human body. And what are the sources of these sugars and fibers? It can either be rice, it can either be potatoes, or it can either be fruits. So we have different types or sources of carbohydrates. Now, what are carbohydrates chemically? Carbohydrates. Now, what are carbohydrates chemically? These are either polyhydroxyaldehydes, polyhydroxyaldehydes, or these can be polyhydroxy ketones. Now let's discuss these two terms first because these are the basis of carbohydrates. Polyhydroxy. Polyhydroxy is something that has different H and OH groups. And aldehydes in organic chemistry, we have already studied that what are aldehydes? Aldehydes are something that have a CHO group. This is very important to remember. Aldehydes will have a CHO group, whereas a ketone will have a C double bond O group. These two, 1, 2, 3, 4, these are the four bonds formed by carbon. So ketone will have a chemical composition of C double bond O, whereas an aldehyde will have a CHO. Now I'll give you an example of a basic polyaldehyde and a polyhydroxy ketone. It looks something like this. Carbon and carbon. Here we have OH. Now CHO bond will be formed here. C double bond H. So this shows that this is a polyhydroxyaldehyde. Now, how can we make out a polyhydroxyaldehyde from a polyhydroxy ketone? A ketone will have a C double bond O group. I'll show you how a basic polyhydroxy ketone looks like. It looks something like this. So this one suggests that this is a polyhydroxy ketone. A polyhydroxyaldehyde or a polyhydroxy ketone is supposed to be a single unit of carbohydrate or saccharide. The carbohydrates are also called as saccharides. Why? Because saccharide means sugar and most of the carbohydrates are sugar by nature. In fact, all of them are sugar by nature. Some may have lesser sugar, some may have more sugar, but more or less they have saccharide units. So a basic a basic polyhydroxyaldehyde or a polyhydroxy ketone is basically called as a saccharon, which is the basic unit of carbohydrates. I'll show you a picture for this. So we have here we can see. This is our polyhydroxy ketone and this is our polyhydroxyaldehyde. So how can we make out the difference between the two? Here we have a C double bond O group. This is the ketone group. This suggests that it is a polyhydroxy ketone. And here we have COH, which suggests that this is an aldehyde group. I hope this is clear with everyone. Now, coming back. Okay. Now we'll study about the classification of carbohydrates. The classification of carbohydrates. The basic classification is on the basis of the saccharide units. As I have already told you, what are saccharides? The saccharides are the basic unit of a polyhydroxy aldehyde or a polyhydroxy ketone. Single unit. Now, based upon these units, classification of carbohydrates, here we'll be studying the classification of carbohydrates based upon the saccharide units. Okay, so on the basis of the saccharide units, these can be carbohydrates can be either monosaccharides, these can be oligosaccharides, or these can be polysaccharides. Now, let's compare the differences between them. First is monosaccharide. As the name suggests, it will have only one saccharide unit. It will have only one saccharide unit. Oligosaccharides will have 2 to 10 saccharide units, and polysaccharides will have more than 10 saccharide units. Okay, now these monosaccharides are the simplest carbohydrates. Simplest carbohydrates. What does that mean? It means that these cannot be further hydrolyzed. Whereas oligosaccharides, since they have 2 to 10 units, these can be hydrolyzed. These are complex structures, so they undergo hydrolysis. Similarly, polysaccharides are also complex units and they will also break down into saccharide units. One polysaccharide can break into more than 10 units because these are made up of more than 10 units of saccharides. So when they break down, they will give us at least 10 monosaccharide units. Then monosaccharides, these are very highly soluble in water, whereas oligosaccharides, these are less soluble in water, and polysaccharides are almost insoluble in water. Okay, now for example, we have glucose, it is easily soluble in water. We have starches, they will take some time to get dissolved, and then we have very complex polysaccharides which do not dissolve, which are almost insoluble in water. Monosaccharides are the sweetest of all saccharides, and oligosaccharides are less sweeter in taste, whereas the polysaccharides are tasteless in nature. So I hope the classification of carbohydrates based upon the saccharide units is clear with everyone. Now there are different kinds of classification of carbohydrates. I'll just show you. Here we have this classification, the basic classification of carbohydrates. So here we can see, based upon the functional groups. This was the basic classification on the basis of saccharides, monosaccharide, oligosaccharides, and polysaccharides. Now, monosaccharides can be further divided into functional group or the number of carbon atoms. Oligosaccharides have disaccharides, trisaccharides, and tetrasaccharides, whereas because they have 2, 3, and 4 units, and polysaccharides are more than 10 units, these can either be homopolysaccharides and heteropolysaccharides. Now, I would like to add here that in this part, in this video, we'll be only discussing upon the about the monosaccharides. The oligosaccharides and polysaccharides, I'll be making a next video for that. So monosaccharides, there are two types of monosaccharides based upon the division or the classification of functional groups. The carbohydrates can be either aldoses or ketoses. As we have already studied, aldose will have a CHO group, whereas a ketose will have a C double bond O group. Also, they can be divided upon the number of carbon atoms. Trioses will have 3 carbons, tetroses will have 4 carbons, pentoses will have 5 carbons, hexoses will have 6 carbons, and heptose will have 7 carbons. So we'll study all these in detail. So let's start with that. Okay, first of all, we'll be studying about what is the chemical nature of monosaccharides. The chemical nature of monosaccharides. Now, monosaccharides, as we know and as the name suggests, have only one sugar unit. So based upon this chemical structure, these can either be crystalline, these can either be crystalline, or these can be easily soluble in water. It depends upon their chemical nature. Now, upon this chemical nature, these can be divided into two parts. These can either be furanose. These can either be furanose or these can either be pyranose. Now, what is the difference between a furanose and a pyranose ring? A furanose ring is basically a 5-membered ring like this. A 5-membered ring. Example of a furanose ring is ribose. That's found in the plants. So here we have an oxygen molecule. We have carbon 1, carbon 2, carbon 3. Here we have carbon 4 and carbon 5. So how the bonds are arranged here? The bonds in furanose ring or ribose are arranged in this way. CH2OH and hydrogen, hydrogen, hydrogen. So furanose ring is basically a 5-carbon ring. Then we have a pyranose ring. A pyranose ring is a 6-carbon ring. Now, the example of a pyranose ring is glucose, the most basic carbohydrate. So a pyranose ring looks something like this. A glucose ring looks something like this. And here we have carbon 1, carbon 2, carbon 3, carbon 4. Here we have carbon 5 and this is carbon 6. So how are the bonds arranged here? Like this. This is the basic structure of a glucose, or we can say this is the basic structure of a pyranose ring. Okay, so on the basis of chemical structure of monosaccharides, the monosaccharides can either be classified as a furanose like this and a pyranose ring. I'll show you a diagram for this. So here we have a furanose and a pyranose ring. Here. So here you can see this is the pyranose ring. The pyranose has a 6 carbon structure. A furanose has a 5-ring carbon structure. So here you can see the oxygen is linked to carbon 1 and carbon 5, and here the oxygen is linked to carbon 1 and carbon 4, and here it is linked to carbon 1 and carbon 5. So this is the diagram for glucose and this is the diagram for a ribose. So I hope the difference between a pyranose and a furanose ring and also the chemical structure of monosaccharides is clear with everyone. Okay. Also here I would like to add something that as we have studied glucose here, a glucose exists in two forms, not just glucose, but different carbohydrates exist in different form. These can either be alpha form or these can be beta form. Now, what is an alpha and a beta form? So an alpha form is basically, or an alpha sugar is basically a sugar in which the OH group is present on the downside, whereas a beta sugar is the sugar in which the OH group is present on the upper side. I'll show you what does that mean. For example, this is our, for example, this is our pyranose ring, and the basic pyranose ring, as we know, is the glucose. So here we have oxygen. So we have carbon 1, 2, 3, 4, 5, and 6 here. Similarly, we have carbon 1, 2, 3, 4, 5, and 6 here. Now, the basic structure is of glucose looks like this. We have hydrogen, 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 CH2OH and H. Now, this is the alpha form of glucose. Now, how do we differentiate an alpha from a form from a beta form? The whole structure would be as it is. The only difference lies on the position of carbon 1. Here, OH is downwards and H is upwards. So, this is the alpha carbon. But in beta glucose, or a beta sugar, what happens is that this bond is reversed. C1 will have OH on the upper side, H on the lower side, and rest all of the structure remains as it is. So this is called as an alpha sugar and a beta sugar. An alpha sugar will have OH on the downside on carbon 1, and a beta sugar will have OH on the upper side, on carbon 1. The position will only vary upon the basis of carbon 1. So I hope this is also clear with everyone. This is also one of the most important chemical properties of carbohydrates, which is called as stereoisomerism. This is also called as the stereoisomerism. So let's get into detail for stereoisomerism. Okay, now stereoisomerism in glucose, because we are studying about monosaccharides, so we'll be taking up the glucose. So as we have already discussed that the physical or the functional stereoisomerism is something in which there is a difference between the structure of an alpha and a beta sugar. Based upon this stereoisomerism, we have different kinds of stereoisomers. We can have epimers. Okay, so what are epimers? Epimers are those are isomers, sorry, which have different arrangement of H and OH group only at one asymmetrical carbon. Now, what is an asymmetrical carbon? An asymmetrical carbon is a carbon which will have different bonds which are bound with carbon. For example, in glucose, I'll show you a chain structure of glucose. The chain structure of glucose looks something like this. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. This is the open chain structure of glucose. Now, if here we see carbon 2, see carbon 1 has different bonds because this one is the aldehyde group. This shows that this is polyaldehyde. But with C2, we can see it has different bonds. Similarly, C3 has all 4 different bonds. Similarly, C4 also has 4 different bonds. C5 will also have 4 different bonds. So C2, C3, C4, and C5 are called as asymmetrical carbons. What are asymmetrical carbons? An asymmetrical carbon is a carbon which will have all 4 different bonds. So C2, C3, C4, and C5 are basically the asymmetrical carbons that we have. Now, what are epimers? These are the stereoisomers which have different arrangement of H and OH groups only at one asymmetrical carbon. I'll give you an example of an epimer. The epimers are D-glucose, are D-glucose and D-mannose. I'll show you how the epimers look like. The epimers. So here, when sugar differs from one another in only one chiral center. What is a chiral center? That which has an asymmetrical carbon atom. An asymmetrical carbon atom is the atom which will have kinds of four different kinds of bonds on all the four different structures. So here we have, this is D-glucose and D-mannose. These two are epimers of each other, which means that they will have same arrangement of H and OH except for one carbon. See, if we see this carbon, H on the left side, OH on the right side, similar is here. Here also we can see H is on the left side, OH is on the right side. Here in D-mannose also it's the same. In D-glucose, we can see on third carbon, OH is on the left and H is on the right. And D-mannose also, it's the same thing. Now, where does the difference come? Here is the difference. On carbon 2. In carbon 2, D-glucose has H on the left side and OH on the right, whereas in D-mannose, OH is on the left side, whereas H is on the right side. So, we can say that D-glucose and D-mannose are basically the epimers of each other. So I hope epimerism is clear with everyone. So after epimerism, the next we have is diastereoisomerism. What are diastereomers? Diastereoisomers. So diastereoisomers are those groups which have different arrangement of H and OH groups at more than one asymmetrical carbons. They may have the difference in maybe 2 carbons, 3 carbons, 4 carbons. Means in more than 2 carbons, there will be the difference of H and OH. Arrangement of H and OH will be different in more than 2 carbons. These are called as the diastereomers. The example is D-glucose and D-aldose. You can take your own examples also, but these are the very basic carbohydrates that I am comparing. So diastereomers, I'll show you a picture for this, for diastereomers. So here we have the diastereomers. See, D-aldose and D-glucose. We have on carbon 1, we have the aldehyde group. Now you can see the difference. There is a difference of H and OH on carbon number 2, on carbon number 3. 4, 5 and 6 are the same, but there is a difference in the arrangement of H and OH on carbon 2 as well as carbon 3. You can see you can notice the difference here in carbon 2 and carbon 3. So since this has a different arrangement of H and OH at more than one asymmetrical carbon, so these are called as the diastereomers. Then next is enantiomers. The last kind of stereoisomerism in monosaccharides that we are going to study is called as the enantiomers. So let's take up that. Enantiomers. Now, in enantiomers, first I would like to tell you something about the D and L configurations of sugars. Now, what is the D and L configuration? D is called as the dextrorotatory configuration and L is called as the levorotatory configuration. So what are dextro and levorotatory sugars? Dextrorotatory sugars are those sugars which when dissolved in a solution, we'll dissolve them in a solution, then we'll pass an optical light through them, and if they deviate this light to the right direction, it is called as dextrorotatory. And those sugar solutions which will deviate the light into left position are called as the levorotatory sugars. Okay, so basically the enantiomers are basically D and L configurations of a sugar, or in basic words, we can also say that enantiomers are those stereomers which are non-superimposable mirror images of each other, which means when we open them into chain structures, when we make a chain structure like these Fisher projections of them, they will not be superimposed upon each other. Such sugars are called as the enantiomers. The basic example is alpha and beta glucose. Alpha and beta glucose structures are enantiomers of each other. I'll show you a diagram for this. So the structure of enantiomers. Here, can you see? This is the D glucose, this is the L glucose or the alpha and beta glucose. See, here we have HOH and HOH. This is a superimposable image. Here we have HO and H, here we have HOH. HOH and HOH. These four carbons, 1, 2, 3, 4, are superimposable mirror images. Now, where does the difference come? Here is the main difference. Here, this is the carbon 1 basically. HOH, this is carbon 1. Here, what happens is that on carbon 1, we have OH on right side, whereas in L-glucose, we have OH on the left side. So because of that, these two cannot be superimposed upon each other, and these two are the non-superimposable images of each other. So such sugars which are non-superimposable upon each other, when they are converted into a chain structure, are called as the enantiomers. So one more thing I would like to add is that these L sugars, these are lab sugars. These are made in labs. These do not occur naturally in nature. Whereas the D sugars, for example, the glucose that is found in the human body, the D sugars are natural sugars that exist in human body or that exist in nature. So I hope this is clear with everyone. I hope this monosaccharides part, the basic introduction of carbohydrates as well as the monosaccharides is clear with everyone. If there is anything that you want a detailed description about, or if there is any topic that you want in detail, you can always write to us. Your feedback is most welcome. And in the next video, we'll be discussing about the oligosaccharides and the polysaccharides. So hope to see you all in the next video. Thank you.